June 19 variant 3 paper 6. paper 6 بقى ما بتاخدش وقت. doesn't take time. هي نفس scheme of questions as we solved before. Figure 1.1 shows a section through an unfertilized chicken's egg. The egg is made up of the outer shell, inner yellow yolk, and albumin. The albumin and the yolk are composed of different substances, including fats. Describe how ethanol can be used to test a sample of food for the presence of fats. To test for fats, we add the ethanol, okay, and we add the fats. Take a sample of food, add ethanol. When the food dissolves in ethanol, it will be clear, and then we add water. If there is fats, it will turn cloudy. If there is fats, it will turn cloudy. Okay, but this is the food test for fats. A student was given a sample of albumin. They tested the sample for reducing sugar. We put 2 cubic centimeter of albumin and then Benedict reagent, and then they were mixed. So there is a step missing for the reducing sugar test. After I mix, do the color appear automatically? No. You have to do an extra step, which is a heating in water bath 80 degrees Celsius. The Benedict test does not work without heating. You have to heat in 80 degrees water bath. Okay. What is the positive result for the reducing sugar? Easy. Color changes to orange, changes to red, changes to green. It changes from blue to any color. It's correct. Yellow, green, orange, red, anything. Okay. He completed the experiment. He told you that proteins can be broken down by protease enzyme. Enzymes are also made of proteins. The student investigated the effect of acid on the breakdown of albumin. I want to investigate the effect of acid itself, not the concentration of the acid, no, the presence of the acid. Okay, so this is the changed variable. This is the changed variable. I want to see, does the albumin, how the protein breaks down by this enzyme, is it affected by acid or not? So I got 3 test tubes. All of them had the substrate. Here I put water only. Here I put 1 water, 1 enzyme, and here I put 1 acid and 1 enzyme. All of them were placed in water bath 40 degrees Celsius for 10 minutes. This is a controlled variable. However, it's a source of error. Why is it a source of error? The error will be temperature is not maintained, which means that if I leave it, it will definitely cool down to the room temperature. So it's not maintained, it will change. Okay. After 10 minutes, the test tubes were removed from the water bath, placed in a test tube rack. 2 cubic centimeter of Biuret solution was added to each test tube. The appearance of solution in each test tube was observed. Anything with colors, we said that is difficult to judge by the eye because it's subjective, so we have to use a colorimeter. He gave you the results, the observation, the student's observation. Test tube P gave me purple color, which means that the protein is still present. It was not digested. Test tube Q, protein still present, gave me purple color. R is the only one that is blue. Why is this? Because it had the enzyme, it had the acid, and as we know, pepsin enzyme, which digests proteins, requires pH 2, requires an acidic pH to work. Type. The first question is, prepare a table to record your results. As a rule, the changed variable is on the left. The results is on the right. طيب أنا I do not have an exact numbers for the acids. He did not مثلا change the concentration. No. He changed the contents of each test tube. So we will just write the name of the test tube, PQR, The observation is purple, purple, blue. What is the conclusion for these results? A conclusion. What's the conclusion here? The conclusion will be P and Q contains protein because it gave me the purple color, while R has no protein. So what is the variable changed? What did I change? I changed the acid or the pH. One of them had acid, the other two did not have acid. I changed the pH. State the purpose of test tube P. If you go back to P, he put the albumin and water only, no enzyme. When he removes the condition under investigation, we say that this is a control experiment. Control experiment. Why did I do it? To make sure we write it as 3 points. This is a control experiment to compare results and to make sure that the acid and the enzyme are needed for the breakdown of protein. I need to make sure that the enzyme is the thing that breaks down protein in the presence of the acid. So I removed them both. I found out that nothing happened. The protein is there as it is. Suggest why 1 cubic centimeter of distilled water was added to test tube Q. Why did he add 1 cubic centimeter to test tube Q? يعني in the first test tube, he put 2 albumin and 2 water. In the second test tube, or in the third test tube, he put 1 acid and 1 enzyme. Why in the second test tube did he put 1 enzyme and 1 water? Why didn't he just put this and leave this empty, 0, 0? لا, it will not work this way. The total volume of the 3 test tubes must be the same. Here the total is 4. Here the total also must be 4 cubic centimeter. Here the total must be 4. So instead of adding the acid, he added water, which is the neutral pH, so that the concentration is not affected. Okay? يبقى why did he add 1 cubic centimeter of water? To keep the total volume or the concentration of enzyme the same. This is a common error that we saw a lot. The student used only 1 syringe to prepare the solutions. He used 1 syringe to prepare all the test tubes. This is, of course, a source of error because it will cause contamination. You can take some of this solution from P and mix it with Q or Q with R, which will cause dilution of the solutions. You will mess up with the concentration of the solutions. So I don't want this to happen. But this is the error, cross-contamination. It will cause dilution of solution. Dilution of solution. Identify one safety hazard. Take care. He's asking you about the hazard, not the safety precaution. Not the safety precaution. What is the hazard here? يبقى الهازard in this experiment is using acids. Using acids. Okay. Question 1 is done. Let's check question 2. He got me a photo of an artery. Okay. Photo of an artery, and he asked you to draw the cross section of an artery. We did it before. It's an oval shape. Here I have a darker gray layer, which is a bit thin, then a light gray layer, which is thick, and the inner lining is folded. So I will draw a thin outer layer, just lines. I don't need any shading, any coloring in the middle. And then the layer after is thick and folded from the inside. This is the lumen of the artery. This is how you should draw it. Okay? This is the drawing. Do not draw individual cells. He said a large drawing. I do not need labels here. 4 marks. 1 mark for a clear large drawing. Okay? 2 marks, sorry, for clear outline and large drawing. And then 1 mark for the layers. The 4th mark is for the wrinkled edge. Wrinkled edge. Of course, after the drawing question, he gives you a magnification question. Figure 2.2 shows a photomicrograph of an artery and a vein. The diameter of the artery is indicated by the line AB. Put your triangle, magnification equals image over actual. Measure the length of AB, include the unit. You will measure it with the ruler. It will give you the length in centimeter. Multiply by 10. It will be 37 millimeters. Centimeter is also correct, but I prefer using millimeter in paper 6. Calculate the actual diameter. I want the actual, so I will put image over magnification. Image over magnification, okay? 37 over 13. Where is the magnification under the photo? 37 over 13, 2.846. To the nearest two significant figures, you will take the first two numbers, so it is 2.8 millimeter. If this is calculated in millimeter, the answer will be in millimeter. If it is calculated in centimeter, then the final answer will be in centimeter. Both are correct as long as you write the unit. Describe one similarity, one difference between the artery and the vein. The similarity is something that is present in both. I can say that both have layers, both have lumen, okay? Both have the same number of layers, both have 2 layers. All of these are similarities. But what about the difference? Here I have wide lumen, here narrow lumen, here I have thicker wall, thinner wall, okay? The size of the lumen, the difference is the thickness of the wall, the size of the lumen, okay? Okay. A person investigated the change in their pulse rate before and after exercise. He measured the pulse rate before exercise, during, and after. Calculate the percentage increase from minute 6 to minute 12. From minute 6 to minute 12, calculate the percentage increase in the pulse rate. How do I do it? Final number minus original over original times 100. Where is the final? He told you from 6 to 12. So at 12 minutes, this is the final. 6, this is the original. يبقى I will do 160 minus 78 over 78 times 100. He wanted it to the nearest whole number, so it will give me 105. Plot a line graph of the time against the pulse rate. He wants a line graph of the time against the pulse rate. Left side of the table is always on the x-axis. The results you are measuring is on the y-axis. This is the dependent variable. The dependent variable you are measuring is on the y-axis. طيب, since the numbers are written at equal interval 2, 4, 6, 8, 10, then you can write them as they are on the x-axis scale. What about here? The smallest number is 78. The biggest number is 160. You do not have to start from the 0. You just have to put a scale that covers these numbers. So my scale can be from 60 to 160. I will cover all these numbers and I will cover more than 50% of the graph. The first thing to do, put the labels time / minutes 0, 2, 4, 6, 8. Put dashes to know exactly where is the number. Okay? On the y-axis, pulse rate beats per minute. 60, 80, 100, 120, 140, 160. And then you start plotting your points. The first 3 points were at 78. Where is the 78? In order to calculate the value of each box, calculate the difference. It's 20 divided 10 boxes, so each box is for 2. يبقى this is 78, 78, 78. and then 125, and then 140 something, 148, 160. You put all the points. After you put the points, you join point to point using a ruler. In the line graph, join point to point using a ruler. If you did it with your hand, it will be still correct, but you have to pass through the center of all the points. Do not miss any point because it's not the best fit. Use your graph to estimate the pulse rate at 15 minutes. How am I going to estimate the pulse rate at 15 minutes? From your graph, you will get your ruler. You will draw a line and the 15 minutes go up at 15 minutes until you meet your graph. Okay, 15 is exactly in the middle between 14 and 16. Okay, and then when you meet the graph, you will go left. مثلا for me here, it's around 138 beats per minute. For your different graphs, this might be slightly different number, 139, 140. It will still be correct if you read correctly from your graph. You just have to show him how you got the number. The question after is a describe question. Describe the results. Description. The pulse rate was constant and then it increased until minute 12. It reached the maximum of 160 beats per minute at minute 12, and then it started to decrease. It did not return to the normal level, to the resting before exercise. You can also use numbers. Use numbers from the graph. How am I going to use numbers? At 8 minutes, it was 122, while at 12 minutes, it was 160. So calculate the difference, subtract them, and tell him that it increased by 18 beats per minute. Sorry, 38. 38 beats per minute. It increased by 38 beats per minute. The last question is the plan an investigation to find out how running at different speeds affects the pulse rate. How running at different speeds affects the pulse rate. What am I going to change here? I want to find the effect of running at different speeds. So this is the changed variable. I'm going to change the speed of running. I have two options. Either you will get the same person and you will make him run at different speeds, or you can get 5 people, make them run at different speeds, but the 5 people has to be same age, same gender, same level of fitness. Okay? Okay, for me here, I used one person. I will measure his resting pulse before exercise, and then I will make him run at different speeds. Okay, this is the changed variable. Type. What is the controlled variable? You got the same person, so this is a controlled variable. The temperature must be the same every time he runs. The temperature of the environment around him must be the same. The clothes he's wearing must be the same. Everything must be the same. Okay? Same distance of running. You change the speed, but he will run for the same distance. Rest for the same time in between the speeds. He will start slow and then measure the pulse rate after exercise, and then he will rest, and then he will run faster, stop, rest, run faster, stop, rest. He has to rest between the speeds, so the pulse rate returns to normal. Number 4 is always your results. What are the results you're measuring? Measure the pulse rate immediately after exercise. Now, what is the safety precaution in any experiment with safety precaution? With exercise, you have to wear proper running shoes and then repeat at least 3 times and calculate an average.